Hello everyone. So the other day I posted a video about where I got some algae and I did a count on the algae, and I kind of explained how it worked, but I didn't really go in depth into the actual mechanics of how it works. Now I'm going to be doing another algae count. So this time I'm going to do the count, show that count to you, and then I'm going to explain how it works, including the mathematics. So let's get started. So the way you do a count, any cell count, whether it's sperm count, blood cell count, or algae or any sort of count, bacteria, is you use something called a hemocytometer. It's this glass piece right here, and the video thumbnail has a better close-up of what it is. But essentially, it has two squares on it, and the two flat squares have laser etching, which I'll show you later what it looks like. The squares, you can see them under the microscope, and you count the cells in the squares. And the way you do it is first you put the slide cover on there. And the way this hemocytometer works is it's a special slide cover. And it isn't flat. It's not like a regular slide where you drop it and it just smears out flat. There's raised edges where it keeps the slide cover just above the those etched squares. That way, only a certain amount of fluid can actually fit on inside that square. And I have my sample right here of algae that I took from the incubator. I'm going to do 10 milliliters each on each side. Now there's two sides to this counter, where you have two sets of etched squares. Now you don't need to use both sides, and theoretically you can use each side for a different sample as long as you don't mix them. But I like to use both sides simply because that gives me, instead of using 4 squares to count, it gives me 8 squares, which gives me a statistically more accurate count. Essentially, you get a larger sample size. Because the way you're doing this is it's a count, and based on essentially on statistics. There's 1/10,000th of a milliliter essentially underneath the slide, and by scaling that up, you can tell the concentration per milliliter. Now I gotta try to. There it is. Now hopefully you can see the squares right there. And what I use is this thing, which it's one of those clickers. And the reason I do that is so that once you get into a large cell count where you have possibly 80, 90, 100 cells in a square. Here right now it's a very small amount because I just got the algae in and it hasn't had time to grow much. But once you get to 80, 90 cells, like there's no point in trying to keep track of it and writing it down. You want to get this done quick. Now right here I don't see any. There's one. Now this one is outside of the square, which I'll explain later, so I don't count it. I'll have to focus here. There we go. There's one. Now as you can see, there's not that many, so. And we got a count of 1, which I could get a more accurate count, which I might try to do by essentially undiluting it, so to speak. I'll explain to you exactly how this works. So, as you saw with the etching, obviously it's magnified, so you couldn't see the whole thing. This is essentially what you see, or if you were to magnify it out enough, this is what you would see. The way it works is each one of these squares is a millimeter, and then it's broken down by a quarter millimeter, and then you can get each square, as you can see, it's dependent on the magnification. So obviously if you have a higher magnification on this, which I believe I was using 4 times 40, 160. The higher the magnification, you might want to use this, because this set of squares instead of these 4. But what I did is I used these 4, and I used the top and the bottom because it has This etching, there's two sets of it. If you have a low magnification, you could use these, or if you have a really high magnification, you could go down and use, let's say, 4 of these. In which case, in this case, you would use a random set, so maybe 1, 2, 3, 4. So let's say we look at this one square. Let's say you have this square. Now, the way I do it is, if the cell touches the right side or the bottom side, I don't count it. The way you see it here, you have all these cells. You would go through and count. Now, this one, because it's touching the top, I count it. Now, here's the thing. So I went here and I counted this one, and this one. Then I counted this one. Click. And then I didn't count this one, right? Well, when I go to the next square, I count it because now it's on the right side, or now it's on the left side. And then when I go here, even though I didn't count this one, now that I go here, I'd count 1, 2, 3, 4, 5. Now I would count this one, 6, 7, and I would continue on until you made it to a square, to the end squares. So you would count these, and then you go to this one, and you do the same. And in my case, I did the 4 on the top and the 4 on the next set of etching squares. And now that you have, and then eventually what you do is you'll get your count. Now in this case, I did a count of 8 squares. So I did 4, 1, 2, 3, 4, and then on the next hemocytometer, or yeah, the next set of squares, I did 1, 2, 3, and 4 again. So that gave me 8. Now, this is the equation that you use to figure this out. Now, there's different variations of the equation depending on how they want to write it, but essentially it's all the same. N is the number, so you take n times 1 times 10 to the 4, which is 10,000, and that brings it up to a millimeter, right? Or milliliter, because it's a 10,000th of a milliliter. You multiply it up to a milliliter, right? N equals 1. So this makes the math pretty easy. Now, S is the number of squares, right? So S, in my case, would be 8. Now, D is the dilution factor. As you see up here, there's a lot of cells. Imagine if those cells were really, really packed. It'd be very hard to count. So one thing you can do is you can dilute your sample and then work it out in this equation. Now, so let's say your original sample is 5 milliliters. What you can do is, let's say, if you want to cut it down by 1/5 to get a 5-to-1 dilution, you would, to that 5 milliliters, you would add 20 milliliters, and that gives you 25 milliliters. Well, because this is a 5-to-1 dilution, what you would put in here would be 5. And so it would be n times 10,000 divided by S times D, which would be 5. Now, in my case, because I didn't dilute it at all, D equals 1. Now, I put this in here. Normally, this is where it ends. This will give you the cells per milliliter. The reason I have this in blue is because this is something that I do especially for my my lab, my experiment. This is the volume in milliliters. So in my case, I have my algae sits in a 500 milliliters of water. That's the whole volume of the sample. So in my case, the V equals 500. Now, because I want to know how many cells, or how many algae cells or trichomes, they're actually multicellular. How many trichomes? Because I want to know how many total trichomes there are, I have to take the trichomes per milliliter and multiply it by how many milliliters I have, and that'll give me the total population of the entire culture that I have. Okay, so n times 10,000 is 10,000, and then you divide it by 8, which is the S, So the actual number is 1250, okay? 1250 times 500. I am all messed up today. 0, 0, and then 0. I've been in the lab way too long. Okay, so that's 625,000 cells in the, or trichomes in the incubator. And you can do this with blood cells, sperm cells, whatever kind of cells you need to do counts on. Now what I'll do is I'll log that count into the book, and then keep that record. So that's essentially how you use a hemocytometer. And thank you.